Hi there, welcome to another tutorial by My Lab with Lucy. Today we'll be looking at ELISA, which is an immunological technique, and our overview is going to comprise of the principle of ELISA, the types of ELISA, and a procedure overview, or what I'm calling an ELISA checklist. And ELISA is a biochemical immunological technique. And immunological here now will mean the reactions between antibodies and antigens. And in full, ELISA stands for enzyme-linked immunosorbent assay, or sometimes we call it enzyme immunoassay, EIA. And therefore, the main components of this, of course, is the enzyme linkage, then immunosorbent, because we are having the utilization of antigens and antibodies. So we detect either the presence of an antibody or an antigen within a sample, and this sample can be dynamic. It can be ranging from diagnostic medical samples, it can be plant pathology samples, it can be food industry samples as you're doing quality control checks, because if you have an antigen, you can raise an antibody against it. And then, of course, the antibody that you raise against it can be conjugated to an enzyme for detection mechanism. Once the enzyme will catalyze what you call a substrate, as we shall be seeing in the next few slides. So ELISA is a dynamic and test that actually has been in existence for a long time, and we are using it a lot. And today we also are having what we are calling ELISA based assays. Like you're having a diagnostic kit like this one, okay, this diagnostic kit here, and it is using enzyme linked immunosorbent assay principle, such that we are having the coating of this diagnostic kit here with what we call maybe an antigen or an antibody against a certain disease. Let's say it's a bacteria or it's a viral antigen, and then now we are expecting either to find an antibody within the patient's serum, and then after that, of course, there'll also be what we are calling now conjugated moieties and molecules that now will again bind to our analyte of choice. And now the conversion of now enzyme catalyzing the substrate gives us a color. And in that case, in this case, for example, being a pregnancy test, it is going to give us lines, and these lines are chromogenic, or they are colored. Okay, so enzyme linked immunosorbent assay is a dynamic test that is being applied in many. If I think I should actually put a card here for the project that I did for my MSc, because it involved the use of, you know, antibodies against a protein called E6 that is found on cervical cancer tissue. Once we are having, the early progression of cervical cancer infection or progression, and I have used now what you call ELISA based principle. Okay, because I did an antibody that was conjugated to HRP, and then I was adding now substrate called TMB for color development, so that now I can visualize positive reactions and distinguish them from negative reactions. So kindly check that card that I'm putting up here. It will help you, visualize what I mean by ELISA being very dynamic. It's being used in diagnostic kits. It is being used now the traditional ELISA test system. Of course, today we are also having kits that have simplified the process, and we also are using it in food industry, plant pathology, among many other areas, even in biotechnology. So the types of ELISA that we are going to look at, the main ones is direct, indirect, sandwich, and competitive ELISA. So the direct ELISA involves an antigen being coated on the well, and this well is called microtiter plate well. So the antigen is going to be coated on this well by passive adsorption, and then maybe let's use the example of HIV. So the HIV antigen is going to be coated on this well, and then we are going to add now a labeled antibody, and this antibody is labeled by adding what we call an enzyme, so that this enzyme helps us in catalyzing a substance that we will add into this reaction to generate a colored signal. Now, if we use this in the context of how you diagnose disease, then it would mean that from the patient, we have collected, let's say, serum that we are proposing to be containing HIV antigens, and that is the serum that we are going to incubate on our plate for several hours at 37 degrees to allow it to bind or to coat onto the wells. And then nowadays, of course, anti-HIV. So this will be anti-HIV, so that now the antibody can identify the target, which is our HIV antigen, and of course now the substrate addition and the production of color. In indirect type of ELISA, we have our solid plate coated with an antigen, and this antigen we can still use HIV. So the HIV viral antigen is coated on our wells by passive adsorption and incubation, and then now we add a primary antibody. So in this case, this HIV antigen is pre-coated or synthetically made, so that now in here we are going now to add our serum, and in this situation our serum is the one that we are proposing now to be having what? Antibodies against the HIV antigen. And then after that, now we will add the detection mechanism, which is now a secondary antibody that is conjugated to an enzyme, and of course addition of substrate for us to generate chromogenic signal. Now if you look at this, in the first situation here, we were detecting from our patient HIV antigens. In this situation here, we are detecting from our patient HIV antibodies. Okay, so you can see that different tests, the way I said at the beginning, you are either detecting an antigen or an antibody. So when you're detecting an antigen, you are raising an antibody against it. When you are detecting an antibody, you are raising antigens against it, which now in this case we have pre-coated on the wells. I hope that is clear. Then sandwich ELISA usually will involve even more number of antibodies to maximize our binding capacity of our analyte. Now our microtiter plate will be coated with what we call a capture antibody. Now this capture antibody is able to bind to the antigen of interest. Let's still use HIV from our patient serum. So in this case, we are detecting HIV antigens from our serum. So this capture antibody is able to bind to the antigen, and then we can do it directly, or what we call now direct ELISA, by having now another antibody that also binds to the same antigen, but now that is labeled with the enzyme. Now, remember we talked about direct ELISA, which was having just the antigen and the coated or conjugated enzyme. And now that is the same case we are saying here. So the only difference is that in this being sandwich that is direct is the fact that we had another antibody that was coated on our well. So this antigen here is detected by two antibodies, but they are binding to different epitopes. Okay, they are binding to different epitopes on the same antigen. Of course, substrate addition so that you can generate color. You can have sandwich indirectly. By now, you already added your analyte of choice from the serum, which is the HIV antigen, and then now you raise a primary antibody that is not labeled, okay, and then you raise a secondary antibody that is labeled with your enzyme. So the secondary antibody, of course, has the enzyme that will catalyze the substrate and finally give us a chromogenic color that can be measured. So I hope that is clear. Sandwich ELISA, capture antibody, target your serum antigen, in this case HIV, then do it directly by adding directly a primary antibody that is conjugated to the enzyme, or do it indirectly by adding a primary antibody that is not conjugated and adding an antibody against the primary antibody that is conjugated now to the enzyme. Competitive ELISA, we have now our wells being coated with an antibody, and now here we add the patient antigen. Let's still use HIV antigen from the sample, and then we add another HIV antigen, okay, that is synthetic. This is from the serum of the patient. This is synthetic and it is conjugated to an enzyme. This is an enzyme. So you add serum for which you're suspecting to be having HIV antigens, and synthetic HIV antigen that is coated to an enzyme. So these two, because they are the same antigens, are going to be competing for binding sites onto the antibodies. And therefore, depending on which antigen is more abundant in our experiment or in this setup, then if it is the patient antigen that is more abundant, it will compete successfully, and therefore the one that is conjugated to the enzyme, once substrate is added, there'll be less signal. If the synthetic conjugated antigen was abundant, it would compete successfully and therefore auto-compete the serum HIV antigen, and therefore there will be more signal. Okay, so this kind of ELISA, competitive ELISA, usually has what we call indirect proportionality between the concentration of analyte of choice versus the color development, such that if you find there is a lot of color, it means our analyte of choice was low. Okay, our analyte was low. In all the other types of ELISA, if you find that the color is more, then it means that our analyte is more. Okay, so for competitive, we have indirect proportionality, which we usually use standard curves to even determine the concentration. But for direct, indirect, and sandwich, we have direct proportionality. Okay, so indirect and direct proportionality. This competitive ELISA can also be done by coating our well with the antigen instead of the antibody, and now adding antibodies that are from the patient's serum and synthetic ones that have enzyme conjugation. These are enzyme conjugated, these are from the patient's serum, and the same case applies. The more abundant one, if it is the one from the patient's serum, that compete successfully to bind to the antigens, then there'll be less color. Okay, if it is the ones that are synthetic and they are conjugated to the enzyme that are more abundant, then there'll be more color. So the different advantages and disadvantages of each of these. So what we need to note is that mainly direct tests are highly sensitive, sandwich are both sensitive and specific, and also competitive are highly sensitive. Okay, because for any diagnostic kit, beyond cost, beyond the time it takes, one of the most important things is sensitivity in terms of how low can we go in detecting our analyte of choice. So if someone is infected with a virus, can it detect even the minute picograms, of the virus in the system? And then how specific can our test be able to distinguish between HIV, hepatitis C, hepatitis B, among other viral diseases, or even among any other molecule that you would want to target, so that we do not misdiagnose our patients. So, in terms of sensitivity and specificity, sandwich ELISA is the best, but of course, it is expensive because it involves the use of primary and capture antibodies as well as secondary antibodies. And then, of course, this indirect sandwich and competitive have an issue of cross-reactivity between secondary antibodies. And another important advantage of direct, of course, it's very fast. And because we don't have use of secondary antibodies, there is less cross-reactivity. In our workstation ELISA kit checklist, we need positive control, we need negative control, we need the wells that have been pre-coated with antigen or antibody, we need primary conjugated antibodies, we need secondary conjugated antibodies, we need the substrate, we need test samples, and we need what we call a stop solution to stop the reaction between enzyme and substrate. For equipment checklist, we need a microtiter plate. This one, we need pipettes. Here they are. We need pipette tips. We need wash buffers. In this case, we are having an automatic washer. So the washer here, we need paper towels. We need marker pens. We need the ELISA reader for now reading the optical densities. And this is an example of an ELISA reader. And then we need an incubator for the incubation steps of coating and all that. And then, of course, gloves, another PPE. Another important thing is that we need a quick guide of the laboratory test kit that we are using, and I will be doing one of them in the next lesson. So the general procedure, I want to highlight 4 things. I probably have mentioned them before. There is what we are calling coating, where we treat our plate with a solution of antigen or antibody that we want to, raise now antibodies or antigens against. And then the areas that do not bind this antibody or antigen in this case, we usually block using bovine albumin serum as one of the examples of the unrelated protein-based solutions. And this helps in reduction of noise or unspecific binding of now our analyte on these sections of the well. And then after that, of course, we have the chromogenic detection where we measure optical density and of course read out the ODs using spectrophotometry. So for chromogenic detection, we utilize enzyme conjugation, and these enzymes will catalyze substrates. So for alkaline phosphatase, the substrate it will catalyze is, for example, PNPP or BCIP-NBT. When you use horseradish conjugation, it will catalyze TMB, it will catalyze ABTS, and it it will also catalyze OPD. And then which depending on which enzyme and substrate you have used, there'll be different colors that are going to be observed, of course, and even the reading of results will be specified in your kit in terms of what is the wavelength to read the color changes. And that is it about the overview of ELISA. If you have any questions, comments, kindly remember you can put them on the comment section. Look out for the next video. I will be doing just a general review of a kit, an ELISA kit that I have used before, and I think it will help you now look at the various specifications that will come with every kit anytime you want to carry out an ELISA protocol. Thank you so much for watching, and I hope you have learned. Let's meet in our next lesson.